Welcome to our last topic for Chapter 3. This is on classifications of prokaryotes. This is a very short topic, so hopefully you'll be able to focus in and get through this last topic for this chapter. So why is this topic important? It's important that we be able to understand the way that bacteria is classified and compared to each other. So that's why it's important that we are familiar with some of the classification systems. So we have two main classification systems. The first of this is the determinative bacteriology manual. This is phenotypic information. What kind of gram stain do they have? What kind of shape? Is it a rod? Is it a cocci? Is it a spirochete, etc.? What kind of growth media does it work in? So this is really stuff that you can easily do in the lab, in our basic bacteriology methodologies. And these are things you're going to be doing all semester. So this is the manual that you would use for this to help identify that unknown bacteria. We've been able to sequence segments of the genetic information of these bacteria. And by using this, we can compare the rRNA sequence data to learn how familiar or how similar bacteria are to each other. This is in the Berge's Manual of Systematic Bacteriology. This helps us be able to compare and contrast, like evolutionarily how similar they are. This has helped us redefine several different groups of bacteria over the last several decades. So it's really important to make sure you understand that. The Berge's Manual is really good if you have an unknown bacteria that you're trying to identify, whereas the systemic bacteriology is good for comparing and contrasting two different microbes through their genetic sequences. There's 4 types of divisions based on the Berge's Manual. We got gracilicutes, firmicutes, tenoricutes, and mendocicutes. I'm not going to ask you to be able to regurgitate these, but I do want you to be familiar with them and be able to tell me what the differences are. Note that they are divided by cell wall, just like we've been talking about. We have our gram-negative cells, we have our gram-positive cells, we have the ones that lack the cell wall, and we have the archaea with unusual cell walls. So just be very familiar with these and you'll be all set there. Last thing is, let's talk about a species. So the definition of species arose from talking about animals and plants, where they were species is isolated as something that can breed within itself and create fertile offspring. That doesn't quite hold true in prokaryotes. So instead that we state that a species of bacteria shares at least 70 to 80% of their genes. However, within the species, we can have several subspecies, strains, or types. These are distinguished because they have different characteristics. This can be seen in a whole variety of bacteria. We also have subclass or serotypes, but the difference being that a serotype creates a different antibody response within the host compared to a subspecies, strain, or type. So for instance, an example of a serotype comes from E. coli. Everyone's heard of O157. There's also E. coli O111, for instance. There's several of these, so that they break down into different serotypes. So that's important to understand. There are subspecies, strains, types, and serotypes. I'm not going to ask you to be super familiar with them, but do understand what those basics are. This is as I promised, this is the end of a very short topic lecture. So once again, let me know if you have any questions. I know this can be a little murky. This might actually be some of the harder material for this topic or for this chapter, even though it's short. So please don't hesitate to let me know if you have any questions.